What is the perfect geroprotector? For many years, the main criteria for a geroprotector has been the ability to increase lifespan. In reality, a perfect geroprotector must also have other characteristics, such as the ability to reproduce its effects in various animal models, to delay the development of one or more age-related diseases, to improve the biomarkers of aging, and to cause minimal side effects. A perfect geroprotector must be easy to study, and the effects should be immediately evident. There are two geroprotectors that immediately show all these characteristics in experimental model. They are two pleiotropic molecules that can develop multiple beneficial effects on cardiovascular, neurodegenerative, and metabolic diseases, and are able to slow down and improve the aging process, acting on multiple geroprotective processes, and in particular on mitophagy. I am talking about Spermidine and Urolithin A. The first is a natural polyamine that is known and studied for a long time. Urolithin A is a natural metabolite produced by gut bacteria from ellagic acids. They both improve health and life expectancy in various experimental models, and for which there is an increasing interest for their potential application in regenerative medicine as a nutraceutical tool. But what is quite remarkable is that they can act in a synergetical and complementary manner on mitophagy and mitochondrial dysfunction, which is one of the most important factors behind aging. So, in my presentation, I investigate Spermidine and Urolithin A, analyzing the mechanism of action with particular regards to mitophagy, the studies demonstrating their efficacy and safety, and their application in clinical practice. I would like to thank the organization and Professor Manuel Castillo for the invitation. So, Spermidine is a polyamine compound found in all eukaryotic cells. From a chemical point of view, it is a polycation, so it readily interacts with negatively charged molecules such as DNA, RNA, and lipids. Spermidine was isolated in human semen at the end of the 19th century. It is synthesized from putrescine and serves as a precursor of spermine. Spermidine body pool is maintained by three primary sources: diet, synthesis in the body, and gut microbiota. It has been known that polyamine levels decrease with age. In centenarians, spermidine levels are comparable to young adults. Spermidine is reported to promote lifespan of yeast, flies, worms, mice, and human cells through several key cellular processes, although the beneficial effects on aging are mainly due to the induction of autophagy, and in particular of mitophagy, that is a selective form of autophagy that specifically removes dysfunctional or dense mitochondria to maintain homeostasis and protect against mitochondrial dysfunction. Spermidine leads to mitophagy through two main processes. They are complex if you go into detail. I mention them for two reasons. The first is that some of them are new. The second we'll find out at the end. Spermidine acts as a substrate for the hyposination reaction, so the keyword is hyposination. That is the introduction of hyposine, which is a specific lysine residue modification that activates EIF5A and promotes the synthesis of the pro-autophagic factors, namely TFEB. So the spermidine hyposination TFEB autophagy axis is responsible, at least, of part of the geroprotective effects of spermidine. Second process. Spermidine activates autophagy through deacetylation of specific residues on histone 3, leading to the upregulation of genes related to multiple stages of the autophagy process. Spermidines induce autophagy by inhibiting EP300, the acetyltransferase that directly acetylates and inhibits several autophagy proteins such as TG5, TG7, and LC3. Spermidine may act also through other processes, such as inflammation by inhibiting NF-κB and pro-inflammatory cytokines and modulating FOXO. It reduces oxidative stress. Spermidine carries 4 positive charges, which can function as scavengers of oxygen free radicals, and also it induces antioxidant Gs via NRF2. In addition, it acts as a signal molecule that mimics the state of cellular starvation, activating MPK, inhibiting mTOR, and thus powering mitochondrial processes such as mitophagy and mitochondrial biogenesis. Importantly, spermidine cannot extend lifespan without the functioning of autophagy and mitophagy. Let's analyze the pleiotropic effects. There are so many studies, I'll show you some shorts. As shown in atherosclerosis, spermidine increased the expression of multiple autophagy genes, restoring nitril oxide signaling. Spermidine supplementation normalized aortic wave velocity, a measure of arterial stiffness, and it reduces arterial oxidative stress. Here we can see the polyamine treatment limited in fast sites and improved cardiac function. Polyamine levels are found at elevated levels in the brain, and not surprisingly, its deficiency has emerged as a culprit in different neurodegenerative processes. Spermidine seems to accept its protection against memory impairment and learning capacity via autophagy induction. It is ineffective in Drosophila deficient for ATG7 or ATG8. Recent studies have shown that spermidine is a key factor in the process of adipogenesis, diabetes, and it promotes human hair growth. Let's analyze Urolithin A. That is a natural metabolite produced by gut bacteria from ellagic acids, a polyphenol found in various plant-based foods such as berries, nuts, and pomegranates. In the gut, ellagic acid is converted by the gut microbiota into metabolites called urolithins. We can observe that there are various types of urolithins. Urolithin A is the one with the most healthy properties. Urolithin production depends on individual's gut microbiota, called the metabotype, which defines the individual ability to produce specific metabolites from food precursors depending on the metabolic profile of the gut microbiota. 3 main urolithin metabotypes have been identified. Metabotype A, urolithin A produces, metabotype B, and metabotype 0, urolithin A non-producers. So, after eating foods rich in ellagic acids, only a portion of population is able to efficiently convert this compound into active urolithin A, and those who do have better metabolic health parameters and microbiota composition of the others. Of course, direct supplementation with urolithin A bypasses the need of microbial conversion. The ability to produce urolithin A decreases with age, from 61% in 20 to 50 years old to only 39% in those over 60. The mechanisms of action are different, even if the activation of mitophagy is the main one. Mitophagy is mediated by 2 main pathways, the PINK1 parking-dependent pathway and the PINK1 parking-independent pathway. So the keywords is pathway. The PINK1 parking-dependent pathway is based on phosphorylation of parking by PINK1, a protein kinase that accumulates on the outer mitochondrial membrane of these mitochondria, leading to the elimination of these mitochondria. Urolithin A is able to increase the levels of these 2 key proteins. The PINK1 parking-independent pathway involves mitochondrial receptors that directly bind LCG and trigger mitophagy. There are also other processes. Urolithin A activates AMPK, inhibits mTOR, the physiological inhibitors of mitophagy, increases PGC1 alpha levels, and thus improves mitochondrial biogenesis. It activates SIRT1 and FOXO with antioxidant and anti-inflammatory effects. It reduces inflammation by inhibiting NF-κB and pro-inflammatory cytokines and activating NRF2. It has the ability to modulate oxidative stress, either by enhancing endogenous antioxidant defense or by inhibiting the production of reactive oxygen species. Research has shown its capacity to enhance metrophagy by activating the expression of glutathione esterasperases. Urolithin A stimulates the NRF2 signaling pathway, consequently upregulating the expression of glutathione esterasperases. At the same time of the stimulation of the mitochondrial cleanliness, it also improves mitochondrial respiratory capacity. The effects on aging mediated by mitophagy is conserved for worse to mammals. It extends lifespans and improves half-span parameters such as muscular function. Let's analyze the pleiotropic effects. Urolithin A is negatively correlated with total and LDL cholesterol, meta-inflammation, and BMI, and has a positive potential on cardiometabolic health. It prevents damage and reduces ischemic damage and atherosclerosis in animal models. It improves insulin sensitivity, acting mainly on mitochondrial function in skeletal muscle. This article, published on PLOS Biology, shows that urolithin A exerts anti-obesity effects through enhancing adipose tissue thermogenesis in mice. Urolithin A treatment increases energy expenditure by enhancing thermogenesis in brown adipose tissue and inducing browning in white adipose tissue. Urolithin increased thermogenesis is favored by an elevation of the thyroid hormone T3 in brown adipose tissue. Consistent with this mechanism, urolithin A loses its beneficial effects on activation of brown adipose tissue when thyroid hormone production is blocked by an inhibitor such as propylturacil. Conversely, administration of exogenous tetraiodotyrosine to propylturacil-treated mice restores urolithin A activation of brown adipose tissue and metabolism. Urolithin supplementation has also shown promise as a treatment of senile osteoporosis. It significantly reduces the expression of osteoclast-related genes and bone resorption. This is an exciting study published by Sarah Livingston of the Arizona University that shows how urolithin A and vitamin D cooperate to amplify serotonin gene expression in neuroendocrine cells. Urolithin A enhances the ability of vitamin D to induce the serotonergic enzyme in tryptophan metabolism, namely TPS2, and function as a booster of the action of vitamin D to stimulate gene expression via VDR. Leptin normally inhibits synthesis and release of brain-derived serotonin. Vitamin D represses adipocyte leptin and induces TPS2 to enhance serotonin relay signaling in the cerebral cortex. So, a combination of vitamin D and urolithin A is an attractive potential to raise serotonin in the central nervous system and perhaps improve mood. Research shows that urolithin A significantly increases type 1 collagen expression, reduces matrix metalloprotease 1 and intracellular reactive oxygen species in senescent human skin fibroblasts. It exerts protective effects against UVA-induced photoaging and prevented various morphological changes typically observed after UVA exposure. Neurodegenerative diseases. In neurons of patients with Alzheimer's disease, mitophagy is deficient, leading to the accumulation of dysfunctioning mitochondria. Urolithin A reduces beta-amyloid and hyperphosphorylated type protein. It attenuates neuroinflammation, oxidative stress, and neuronal damage, and prevents cognitive decline. So, the beneficial effects of urolithin A on brain function are associated with multi-target action that include promoting mitophagy and mitochondrial function, attenuation of oxidative stress and inflammation, inhibiting beta-amyloid and hyperphosphorylated type protein, and regulating tryptophan metabolism, thereby counteracting the pathogenesis of Alzheimer's disease. And finally, many findings established a solid scientific foundation for the application of vitamin D to improve muscle health, promote muscle growth, and enhancing exercise performance. Urolithin A may offer potential benefits as a treatment in muscle atrophy, but also sarcopenia or extended periods of immobility. It can stimulate muscle protein synthesis while inhibiting protein degradation pathway, helping to preserve muscle mass and mitigate muscle wasting. It can stimulate muscle protein synthesis, promoting to muscle growth and maintenance. This effect is particularly beneficial for individuals looking to build and preserve muscle mass, such as athletes and fitness enthusiasts. It shows promise in delaying the onset of muscle fatigue during prolonged or high-intensity exercise. By enhancing mitochondrial function in muscle cells, it provides sustained energy support, allowing individuals to engage in longer and more intense training sessions. By suppressing the inflammatory response, it contributes to the mitigation of muscle pain and inflammation. Moving on to human studies. The first human clinical trial with Urolithin A administration was a randomized, double-blind, placebo-controlled clinical trial conducted over 4 weeks in healthy elderly men and women aged 61 and 85 years. Urolithin A showed a favorable safety profile with no side effects, both after a single oral dose up to 2,000 milligrams or after multiple doses up to 1,000 milligrams, with good oral bioavailability at all doses tested, and this is important. This is a very famous study, randomized, double-blind, placebo-controlled clinical trial conducted on 88 overweight and sedentary middle-aged adults, divided into 3 groups: placebo, 500, and 1,000 milligrams of Urolithin A for 4 months. Urolithin A at both dosages significantly improved lower limb muscle strength, and it clearly showed the involvement of all the pathways we discussed earlier. Muscle proteomic analysis revealed activation of pathways related to mitochondrial metabolism and mitophagy, increased levels of active phosphorylated form of Parkin, increased mitochondrial DNA, nuclear DNA ratio as an indicator of mitochondrial content, increased respiratory chain protein in skeletal muscle, a significant reduction in plasma C-reactive protein. This is another randomized, double-blind, placebo-controlled clinical trial conducted on older adults that it is interesting because it shows an improvement in the endurance on both hand and leg skeletal muscles, yet hence the muscles that anatomically and functional are different. Spermidine and Urolithin A are both natural compounds able to activate mitophagy. They both activate the MPK, inhibit mTOR, enhance T2 levels, promote FOX activity, supporting mitochondrial biogenesis and function. Spermidine acts mainly by hyposination reaction, while Urolithin A acts more by activating the Parkin pathway. Their mechanisms of function are different and complementary. At the cellular level, spermidine has been shown to enhance the increase in mitophagy markers induced by Urolithin A. At the molecular level, the combination of the 2 compounds has integrated epigenetic effects. So, combined with physical activity and intermittent fasting, we can consider spermidine and Urolithin A as mitophagy activators. What are the indications and dosages? They can be used as anti-aging compound, of course, for the prevention of cardiovascular, neurodegenerative, and metabolic diseases, for memory, energy, and muscle health. Dosages. For spermidine, the dosage is about 1 to 3 milligrams per day. For Urolithin A, studies report dosages of 1,000 to 2,000 milligrams per day. 250 milligrams twice a day may be enough. Thank you for your attention.